to amino acid. I'm not drawing any ionic forms of it. It will connect to a neighboring compound. Here's another amino acid. One of these H's and that will go away. And these two will connect to each other. That is a condensation between amino acids to form a protein. You've probably done that in biology. This is called a peptide bond. This is all, dehydration synthesis. If you were to separate them, it'd be hydrolysis, water splitting. Okay? Basically, you put water back in. Okay? Pretty basic stuff. Describe and explain. Oh, this is going to be taking me a second or two. Describe and explain the primary, secondary, tertiary, and quaternary structure of proteins. Watch how good I am at this. Watch how good I am. Yeah, for you folks in advanced bio, it'll be a little simple, a little more easy. So, okay, primary. It's the sequence of amino acids. So look, I know it's tough to see the mouse cursor, but here is one amino acid right here. There's the peptide bond. Here's a second amino acid, another peptide bond, another amino acid. This is 3 of them in a row. You're required to know up to 3. Notice that they've still used the R groups. Remember, those could be different R groups. That'll play a role in a second. That's the primary structure, a straight line. These R groups and these other parts of the compound can interact. So these chemicals are sticking off in all different directions. They can attract each other. They can repel each other. That is the secondary structure. The 2 most common ones are called alpha helix. This little curly string is the primary. These dots are supposed to represent side groups. They can interact and cause twisting. That's an alpha helix. This is called a beta sheet, sometimes called a beta pleated sheet. All you have to understand is that they are secondary structure. They come from side groups interacting, repelling and or attracting. Okay? Now, those secondary structures, which are within a strand, those secondary structures can cause different areas on the same string of amino acids to interact. So imagine that this top row is one strand with its interactions, its secondary structure here, and here is a here is the same strand on the bottom far away. So you could imagine that this string of amino acids continues off in this direction and loops back. So this strand acting with itself further down the line, that is tertiary structure. Think of it as a spaghetti strand that is interacting at both ends. It's looping around and both ends end up sticking next to each other. That's tertiary structure within one strand of amino acids. Does that make sense? And quaternary, which means 4th degree, is where you have different spaghetti noodles coming together. Well, I mean, look at it. It looks like this diagram looks somewhat like spaghetti noodle. This portion would be the strand. Notice in some parts it's looped back, so maybe right here you could say there's some secondary interactions here. But further down the line it interacts with itself even more, which would cause a tertiary structure, 3rd level of structure, and then those complex tertiary structures fit with other complex tertiary to make a quaternary structure. It's fairly complicated, but simple at the same time. Any questions on that? Nope. Good. Moving on. Okay, see this one here? Memorize it. Read it. Read it on your own. Major functions, you can read it. Collagen, enzymes, this is online. Hormones, antibodies, just read it online. Okay? Structure, like muscles, easy. Okay, how proteins can be analyzed by chromatography and electrophoresis. Oh, man. You guys are so lucky. You guys are so... Oh, look, there's uses of proteins in this thing. Just memorize it. Okay, analysis. Remember we just talked about chromatography. So, you put your spots on it. You put it in a solvent, they migrate. Very simple. And in this type of chromatography, it has to do with its size and or polarity. Okay? Now, did you guys calculate an RF value before? In your, oops. Okay, this little term right here, this RF value, let me grab that image. Okay. The RF value is unique to each protein. It is the ratio, you can see it here. It's the ratio of the distance the chemical traveled in the chromatogram to how far the solvent went. So in our analogy earlier, how far the river traveled is Y. How far we traveled through the forest is X. Okay? So that ratio of how far the chemical traveled to the solvent's distance, that ratio will be fairly consistent. That is one way of identifying which amino acid it is in a mixture. Does that make sense? And this is simply saying that if you can't separate them from each other this way with one solvent, this is saying you turn it on its side and you run a second one in this direction with a different solvent. And so this spot, these two spots, after this original spot was separated, these are actually mixtures as well. So it was turned on its side and a different solvent was used, which separated them even further. That's all that's showing you. You can read that on your own. Paper chromatography. Electrophoresis. Well, you guys who are in biotech or sorry, I can't do two things at once. Get this out of the way. There we go. One more. Those of you in like a biotech class or even in Advanced Bio 1 doing gel electrophoresis, you have a huge advantage here with this other one. So there we go. There's the zwitterion. Neutral at the isoelectric point. Less than isoelectric point, positive. Above the isoelectric point, negative. So this is supposed to be a DNA gel type thing, a gel box. You put them in, and if the pH of the buffer in this solution is controlled, if it matches this, let's say that the pH is 6.0. Alanine will be neutral at 6.0. But other chemicals, for example, this one, arginine, at 6.0, that's below its isoelectric point. It'll be positive at 6.0. So when you run your electrophoresis at 6.0, arginine will not move. Alanine will not move, but arginine, which is positive, which direction will it move? If the center represents no movement, it'll move towards the negative. Do you understand how that works? It's a method for separating proteins based upon pH. Does that make sense to everybody? Okay. I'm going to stop.